The overall goal of this procedure is to induce mechanical injury of motor neurons in Drosophila larvae.
This method can help answer key questions in the field of neurodegenerative research, such as the temporal sequence of cellular events leading to neurodegeneration. The main advantages of this technique are that it is inexpensive and uses equipment that most Drosophila labs already have.
The implications of this technique extend toward the understanding of the molecular mechanisms that drive neurodegeneration.
To begin the protocol, select 10-second or third instar larvae from a vial or bottle of Drosophila containing wandering third instar larvae that have been cultured at 25 degrees Celsius. Use forceps to carefully pick individual larvae up by forceps or a paintbrush without compressing them. Directly place the 10 larvae into a glass dish containing cold 1x PBS to remove any food debris and to decelerate larval motility. Identify third instar larvae by their anterior branched spiracles, and second instar larvae by their smaller size and club-like anterior spiracles. Place each larva onto a CO2 anesthetizing apparatus. Under a dissecting microscope, carefully roll each larva onto its dorsal side to visualize the segmental nerves throughout the cuticle.
It is essential to carefully roll each larva onto their dorsal side in order to visualize the segmental nerves through the cuticle before injury.
Starting at the mouth hooks, position size 5 forceps approximately 1/2 to